For me, it's a pleasure and is an honor to introduce Dave Stuart. I will give a brief profile of his career. Dave got his first degree in biophysics at the King's College in London. Then he did his PhD and postdoc at the biochemistry department at Bristol University. And then he did a postdoc with Professor Dame Louis Johnson in Oxford, and since then he's remained in Oxford, although he traveled to China several times. And since 1995, he has a post as MRC professor. And now he's also head of the Division of Structural Biology, STRUBI, of the Department of Clinical Medicine. And he's also honorary director of BM14, which is the UK MAD beamline at SRF. And he's the life science director of Diamond, which is the third generation synchrotron in the UK. Dave has been involved in all his career in extremely challenging virus structures. The most important ones, if it's possible to make a very short story, are foot-and-mouth virus, who has caused a lot of economical damage to the UK, but also other countries' cow population, and also blue tongue virus. He has other structure, which I hope we will see today, but these are the, I think, the extremely challenging structure. They are animal pathogens. And Dave is also the coordinator of the SPINE2, which is the structural proteomics in Europe, as well as the Instruct, which is another big project, European project. Now, Dave has been extremely active, and I think in promoting huge realities in the British scientific panorama, but also worldwide. For example, the OPPF, which is the Oxford Protein Production Facility, was fought and pushed through by his scientific view, and is now a reality. It's been a reality for the past 10 years, but now it's moved to Diamond site. Then he has also created what is called the OPIC, which is the Oxford Particle Imaging Center, which is a place where infectious particle can be visualized through a powerful microscope, and as well there is a setup for crystallization in a P3 containment level for high virulent viruses. And finally, I think the biggest challenge has been, I think, Diamond, who has been built in a record time and is functional. Diamond synchrotron is the third generation synchrotron, is very close to Oxford. And Dave has been involved not only in the planning, but right now in managing the scientific side. And just to list some of the prize that has been awarded with the FEBS Anniversary Prize, the Fellow of the Royal Society, the Aminos Prize, which is awarded by the Royal Swedish Academy of Science. He has a Max Perutz Prize from the European Crystallography, and he also has an honorary degree in philosophy as honoris causa by the Helsinki University. So he has more than 270 publications, H number, which I don't know how many digits it has, but his career is stunning. And now I will just give two personal comments on his career, because before coming here, I have been working with Dave and for Dave for almost 8 years. And I use these, I want to highlight the for and with. I never felt in those 8 years that I was working for him. I always felt that I was working with him. Because one of the things that I learned being there is that if you want, you lead by example. And I will never forget the timeless nights collecting data with him, with me at the synchrotron. And people that spend time at the synchrotron knows that between 3 o'clock and 5 o'clock in the morning is a time when frustration comes up, you want to give up everything, and he was there with me saying, come on, let's put these other 20 crystals on in the next 20 minutes. And we were pushing through. This is extremely important, and I value this quite a lot, because science is a privilege. Science, I learned the sense of responsibility, and I think that these are very important characteristics of Dave. You can't complain about working at the amount that you work, because he works double than you. So there was no point for our postdoc saying, oh, we work a lot, because I think that I spend more time with him today as a daylight than in my past 8 years. The only time for him to talk with us was at night, and I think that the most beautiful structure that probably you will see have been solved collecting data at night and looking at the structure at night between 9 and 2 o'clock a.m. And this is the dedication that I learned from him. And the last point is Dave and technology. I always felt that I was a guinea pig in Dave's, because I had always to try the latest technological equipment. For example, the Fluidigm. The Fluidigm is a system for crystallization at very nanoscale, even more than the Cartesian or the nanoscale. And I remember, you have to try with the integrin, you have to try with the integrin, or the Proteus, you have to try. And I felt always that I was doing things that they will never give a result. And that, I think, is the sense of science. You have to try. If you want results, you have to try. And actually, one of the biggest success was for Dave getting a structure of a picorna virus using 1.5 microliter solution, using this Fluidigm. So this is just to summarize my personal view on Dave, and I will leave him now to give his talk, which I'm sure you will enjoy.
Thank you. It's a wonderful pleasure to be here. I'm sorry I can't be here for longer to talk to more people, but I've had a chance to look quickly around, and it's a lovely sight, and I've got the best of the weather, I think. So one of the things that I'm struck by looking around this institute is there's a realization that good infrastructure, good facilities are the sort of cornerstone of doing cutting-edge science. And a significant amount of what I will say in my talk is around technology, I'm afraid. And much of what I'll say is nothing to do with my work, but is really relating to other people's work. But I hope I won't insult people by talking at too low a level about things that you may know much better than me. But I wanted to give you an overview of where I think we are in some areas of structural biology. And I will illustrate it with some examples from our science. So I'm sorry the quality of the images is not wonderful. The colour is a bit washed out. But one of the, as Nicola said, one of the great things about science is being able to collaborate with excellent people. And I just wanted to start off by illustrating some of my collaborators, and I hope we'll be able to carry on collaborating, Nicola, in the future. But in particular, a lot of the recent virus work has been done in collaboration with Dennis Bamford. John Grimes has been involved in many of the projects, as have Carl and Jeff. And Liz is really pushing the picornavirus work, which I'll mention. The picornavirus work that I'll show was also the result of the labours of Abhay Ren and Claudine, shown here on I-24, one of the beamlines at Diamond. What I'll try and go over are some of the new challenges in structural biology. I will very quickly give an indication of the opportunities that arise through new correlative methods in structural biology. And then I will give a little bit more detail about the opportunities for X-ray methods, predominantly X-ray diffraction methods. And then I'll talk a little bit about viruses. And of course, the subtext, the thing that underpins all our vision of the future, is the realization that cells are extremely crowded, busy places, full of macromolecular complexes, many of which are very transient, which are really the machinery by which cells are enabled to operate. So over the next years, the vision for structural biology is to link with cell biology to create an integrated structural biology that will allow you to do what Google Maps does for the Earth, allow you to start off with a view of a cell and drill down to the atomic details within the cell to understand the interactions in atomic detail. That's one aspect, but of course the cell, as I indicated, the cell is very dynamic. There's lots of little different regions within the cell. There's changes all the time. So it's not a simple three-dimensional problem. It's a four-dimensional, an n-dimensional problem to understand the specificity of different tissues. There's no single overall solution to this. It's a very ambitious project. As you'll know by now, there are a range of different methods that we can use to visualize complicated macromolecular assemblies and cells and tissues. And those methods, and I'm talking about visualization methods now, so I won't, I'm afraid I'm not going to go into the opportunities for NMR. They're very major, but outside the scope of what I'm talking about. So if we just consider the visualization methods, we can use light, we can use X-rays, we can use electrons. All of these have strengths and weaknesses. The fantastic achievements of the last few years in pushing light microscopy, in particular using fluorescent probes to resolutions that are well above what you'd expect from the inherent limitation of the wavelength of light, is spectacular. Electron microscopy, again, is making spectacular progress in both pushing down to smaller, more detailed things and pushing up to tomographic imaging of cells. X-rays are sort of span the whole spectrum, obviously from animals through microscopy through to diffraction. But there are real reasons why electron microscopy has big advantages over X-ray microscopy. Not only is it easier to focus electrons than X-rays, but the damage inflicted on biological specimens is inherently less for a given amount of information out of the sample with electrons than X-rays. However, there are also limitations because of the very strong interaction of electrons with matter. You can only visualize very thin sections with electron microscopy. So there's still work to be done to piece together these different methods to be able to get a complete sort of spectrum of imaging modalities. Because the resolution that these different methods provide is very different. So the little blobs here show the precision of the method mapped onto a typical protein structure. So NMR and X-ray diffraction give you atomic resolution. Single particle EM is getting there and advancing in a very impressive way, driven by both technical advances in the electron microscope and also methodological advances in the software. Tomography is lower resolution, but approaching the challenge with electron tomography now is to get the tomography of cells to the point at which it's possible to identify macromolecular complexes and large macromolecules, so that tomography can then be used to produce an atlas where you can label the different machines within the cell. It's not there yet, And there's a big technical and IT challenge to achieve that. X-ray tomography, the sphere of confusion is sort of much bigger. At the moment, the resolution is between 5 and 10 times worse than electron microscopy. So the sort of thing that people are doing then is to try and take biological systems and understand them by putting these methods together. And this is just an example from Kai Grunwald in Struby in Oxford of some of his studies on herpes viruses. So he's able to construct fluorescent herpes viruses. He can then use fluorescent microscopy to follow the life cycle of the viruses in cells, then freeze out particular stages in the life cycle and analyze them at increasingly high resolution using a variety of methods. So there's the imaging methods, but of course, as I said, there's a lot of other methods off-axis, proteomics and so on, that are all part of what you need to do. These advances mean that the technology has become increasingly sophisticated, more expensive, and at some point, and I think we're getting there with the very high-end electron microscopes now, I think we're at the stage where those very expensive microscopes should be run like a synchrotron beamline. This doesn't really happen, at least in Europe, yet, but I think over the next few years there will be a shift to try and do this, to provide a user program and open up these fantastic machines to the whole community. That's one of the things that people in the X-ray crystallography community should remember. It's a very democratic thing, a synchrotron, because anybody, well, at least it is at the moment, as long as we don't lose our sort of nerve and maintain that. At the moment, it's free at the point of access. People can apply and are judged purely on scientific merit. So most of the synchrotrons across Europe are open to people from anywhere in Europe. And that means that the real state-of-the-art technology is available to everybody. And I think we should see the possibility of some of these technologies becoming so expensive that they have to be run as centres as an opportunity to really raise the game for everybody. And that's a sort of sales pitch for this Instruct project, which I'm involved in, which is a pan-European infrastructure programme, part of the S3 roadmap for scientific infrastructure in Europe, which is designed to provide these infrastructures for people across Europe. And Spain is an active part of that. So I'll just give a further illustration of some of these things. This again is not my work. This is just a little quote from a review by Jürgen Plitzko from Martinsried, electron microscopist. And you don't need to read the whole thing, but this is talking now about correlative techniques in microscopy. And it's common sense, okay? The idea is to capture high-resolution snapshots that represent bona fide cellular events. And this is not as trivial as it sounds. If you want to understand how a virus works, you want to be sure that if you take a picture of a virus in a particular context, that you know what biologically is happening at that point. And that's why, using fluorescent probes that light up and tell you about the biology, that needs to be correlated with the high resolution data you get. Otherwise, you can't know what the biological interpretation is. So there's a challenge here, and there's a lot of work to be done, chemistry to be done, to make probes that work for fluorescence and for electron microscopy, for X-rays and so on, so that there can be a genuine correlation precisely between these methods. Then there's the technical problem of if you want to do correlative combined imaging, physically, how do you do it? The simplest thing is combined imaging. And this is the least satisfactory in some ways. You take images of different specimens of the same type, and then you look at them in different methods, and you hope you can correlate the results between them. This is mostly what people do at the moment. And this, again, is just another representation of some of Kai's work. Again, you use various sorts of light microscopy to map out events and then try and capture the higher resolution images. This is the nucleus of a eukaryotic cell. The stage beyond that is to take the same sample and transfer it between instruments. And this is something that it can just about be done, and people can give talks about it, but it's very hard, and just the physical transfer from a cryo light microscope to a cryo electron microscope at the moment is really difficult. So it's not a solved problem in my mind. Okay, but there are now there are several designs of cryo stages for light microscopy and fluorescent microscopy. This is a design that's been produced by FEI. It was designed by the guys in Martin Street at the Max Planck Baumeister's operation. There's also an effective system that comes out of Utrecht. So these exist. They allow you to follow something in a light microscope. They allow you to flash freeze and then go back and check the fluorescence, check the frozen sample to see what you've actually captured. Very, very useful. At the moment, these have limited numerical aperture. They're not the state of the art. There are no immersion lenses that I'm aware of that work that you can immerse in liquid nitrogen. So there's you can't have any immersion optics. So there are limitations, but these are now starting to become a reality. So this means that then you can use this sort of technology to look at plunge frozen cells, and you can do the light microscopy and then transfer to the electron microscope. This works, and there's some results now for this. One of the fundamental problems is that a lot of biomedical work really means that we need to look at eukaryotic cells. And in general, not all of them, but most of them are much too thick to look at with electron microscopy. So what do? Again, you've got a problem here. It means you've got to chop them down. You can section the cells using cryo sectioning. So you section them, these frozen cells. And again, this is possible, but the technology is very fiddly. It's a very skilled operation. And to reconstruct a stack of frozen sections is still very challenging. So this is something which is not routine yet. Another option is, rather than slice them with a knife, is to use a technique called focused ion beam milling, where you send a beam of gallium ions in, which just ablate the surface. So you just completely wipe away the structure. And you can machine down like a little nano milling machine to cut a section of the cell. Now you can see that this then will allow you to produce the right thickness of cell, but you can also see it doesn't take much sort of insight to realize that it's actually very hard to know which bit you look at. You only get one sort of slice. It's, again, a technology that's being developed, but still has problems with ice accumulation, all sorts of things. But here's examples of where it has been used. Here you can see the ablation of these sections so that you can then look at the appropriate activity within here. the sort of ideal in many ways is to look at the same specimen in the same instrument. But this, again, produces further challenges. So at the moment, nobody has combined a super resolution light microscopy with an X-ray or electron microscope that I'm aware of anyway. But there are things that are becoming available, and one of the first developments was from these guys, Bram Kostner and Koster and people in the Netherlands, who've designed, this is the electron microscope, pole pieces, sample on a grid. Here's a light microscope objective. The idea is you turn the sample stage around, bring the light microscope in, you can do the fluorescent microscopy, you retract it, rotate the grid, and then you can do the electron microscopy. So these are, again, at a very early stage of development. At an even earlier stage of development in many ways is full field X-ray microscopy using cryo grids. There's only two, this is best done I think at a synchrotron. There's only two beamlines that I'm aware of in the world that provide this at the moment, one in California at the ALS, one in Germany at Bessie. Only the Bessie one works with modified electron microscope grids, which has advantages because it allows you to transfer over all the sample preparation skills that have been developed over years in electron microscopy to this new field. But there is still an enormous amount to do. In principle, this can be done routinely and quickly. In principle, it should only take a few minutes to collect a tomographic series. In practice, it probably, if you're working for a day, you might collect half a dozen tomograms at the moment. And we don't know whether this method what impact, if any major impact, it'll make on biology. The idea of electron microscopy has been around for a number of years. Cryo X-ray microscopy is much newer. Europe is potentially in a strong position because several other synchrotrons have jumped on the bandwagon and decided they will build these microscopes. But I'm not aware of a sort of killer result biologically that you could achieve in no other way. So this is the jury is out on the role of this in the future. I'll just show this, however, which is unpublished work from Kai Grunwald in Oxford, and this is a tomogram, and you saw a section of it earlier. So again, the representation on the screen is not very good, I'm afraid. You can see the double nuclear membrane around the outside here. So that's the nuclear envelope. And then within it, if you look carefully as you go around, you can see that there are now holes through the nucleus. Now these had been reported before as occurring during a herpes virus infection, but with the X-ray microscopy you can see the whole nucleus. It looks like it's a section. In fact, it's just flat. It's a complete nucleus. So using this method, you can get something you can't get with electron microscopy. You can see inside the nucleus, you can see the whole nucleus at once, and you can see these interesting morphological features, which in fact are sort of tubes, and associated with each of these tubes is a little area here, which is a virus factory. And this is a section through one of the tubes now, going through and segmenting the volume. It's not terribly easy to see, but the tube goes through there, and associated with it is a viral factory where the herpes viruses are being produced and are then sort of being sent out into these tubes, presumably for egress from the nucleus. So I think it's a nice result and indicates there may be areas of biology that could be approached very, very well with the X-ray microscope. But where I've sort of made a living for many years is X-ray diffraction. And one of the questions is, are there major opportunities for advancing X-ray diffraction? And a number of you have probably seen, and if you've seen it, you've probably read in more detail than I have, the papers that came out, I think, last week in Nature on the first results from the X-ray free electron laser. It's the LCLS at Stanford. This is just the tunnel. It involves a very long accelerator section to produce the laser. And those two papers report the result of crystallography on very small crystals, and also report the state of the art on using an X-ray free electron laser to look at an intact virus particle in isolation. The virus particle chosen by Janusz and Hojdu and co-workers was the Mimi virus, and this is cheating because it's as big as a cell, or big as some prokaryotic cells. It's a massive thing, but it's a good test case to look at the method. The nanocrystallography again is really a test case. They took a sample that was already, protein that was already solved at very high resolution to see if they could collect useful data off minute crystals. The setup that's used is similar in both cases. You inject the sample in little droplets of liquid, and then you synchronize those with an X-ray pulse that comes from the laser, and then you have rapid readout CCD detectors to pick up the scattering. So just to sort of, so if you don't know, to tell you how this works, the free electron X-ray laser produces femtosecond pulses of X-rays. Each of these pulses, one femtosecond pulse of X-rays, provides enough photons to give a scattering pattern, which is remarkable. The beam of the X-rays, or the size of the pulse, is of the order of microns at the moment. The present results are using relatively soft X-rays. These are, whatever it is, 8 angstroms wavelength. So there's no way of getting very high resolution structure. In the future, that will change. And I said the sample presentation is rather like an electrospray mass spec. You just shoot them into the vacuum, and you have very clever ways of synchronizing those pulses with the X-rays. Because the pulse is so short, the idea of this is that there's enough photons to completely destroy the sample. But the radiation damage occurs more slowly than the time it takes the pulse to pass through the sample. So the diffraction comes off before the sample knows anything about it, and then it realizes it's dead and it explodes. And you can see that the length of these pulses is somewhere between the time scale for nuclear events and electron events, so it's very, very fast. There's other properties that are not explored fully yet, for instance, the coherence of the beam, and I won't talk about that, it gets a bit sort of technical. But the tests on the small crystals, we're using a photosystem. We're using photosystem crystals. The repetition rate, the frequency of the pulses that come, X-ray pulses, is about 50 hertz. So once you've got a setup there, if you can feed the droplets in 50 times a second, it means that you can collect several million diffraction patterns in a 24-hour period. Each of the diffraction patterns is just a sort of slice through reciprocal space. You need to put all of those together to build up a three-dimensional picture. For this X-ray crystallography, they did this, and they managed to put together a data set to 8 angstrom resolution. At the moment, the data are really awful by the standards of sort of conventional synchrotron radiation. Reliability about 20%, this would be about 2% for standard synchrotron. But of course, these crystals are only maybe 20 unit cells across or 15 unit cells across, so very, very small. So it's a proof of principle, but we don't know at the moment what the potential is. Higher resolution is potentially achievable using shorter wavelengths, and that will come rapidly. You can imagine that a major application, if you can synchronize biochemical events with the presentation of the sample, is for dynamics studies. And that requires, as I say, cunning tricks that people hopefully will develop. If you don't use crystals, at the moment, the number of photons that you can get is not really sufficient to get very high resolution. And this, at the moment, I think is not there as a competitor to single particle electron microscopy. This shows what can be achieved at the moment for mimivirus. These are the pictures of mimivirus. The resolution is that's 200 nanometers, so the resolution is claimed to be, I think, about 200 angstroms. So still not there for a high resolution technique. But if we think about opportunities for whether this might actually open up new things that are otherwise very difficult, this is some collaborative work that we've got going on, but driven by the lab of Dennis Bamford in Helsinki. That lab is working up cell systems which, when infected by particular viruses, instead of breaking open, they remain intact, and the viruses then build up and assemble inside the cell. And these are small and fragile sort of assemblies, but it looks from the electron microscopy data, and UHA in Oxford has now got some tomographic data which supports this, as if there is reasonable order within some of these lattices. And we're trying to do work at the synchrotron to confirm this. If this is the case, it may be possible to completely bypass the normal crystallization procedure and obtain structures of viruses from individual cells if you fire these cells straight into the X-ray free electron laser beam. But that is pure science fiction. But you've got to have some wild idea that you're tackling. So if we go back to standard synchrotrons, don't write them off. There's real developments going on, and those developments are going to have potentially have a big impact on weakly diffracting crystals of complexes, which is, our big challenge. And I'll just remind you, and the people that are interested are probably already aware of these principles. The synchrotrons have tended to be very conservative until very recently in the way they've designed their optics. And over the last years, the machining of X-ray mirrors has improved fantastically. So it's now possible to design a much more ambitious type of beamline. And if this is essentially building a beamline as if it was a zoom camera, right? You know how a zoom lens system works. So you've now got two lenses in here, and you use one set of lenses to produce a secondary source which you can position at will. And the second set of mirrors image that source and focus it at a point of your choice. And this is it potentially makes a massive improvement in the quality of data that can be collected. The example that we've worked with is the I-24 beamline at Diamond. There are very few of these beamlines around yet. But essentially what you can do is your signal, which is the Bragg diffraction, can be focused by this beamline into a very small point on your detector. That then allows whereas the background scatter, which is what introduces error into the signal, is spread evenly over the whole detector. This concentration of the signal it means that the signal-to-noise ratio can be dramatically improved. And these detectors now are increasingly large, which means that the noise is spread out over a very big area and diluted. So but major opportunities here, not yet explored fully, but it's clear that the possibilities are significant. The detectors are brilliant now. People used to use CCD detectors where you would take an image and then you'd have to switch the X-ray beam off while you read out the CCD for a second or so. Now we collect movies on here. At the moment we're collecting movies at about 10 frames a second. So we just switch the X-rays on, rotate our crystal, collect the diffraction data that can give us the structure. Within a year, the repetition rate, the frame rate will be up to 25 frames a second. And each of these frames is 6 million independent measurements. So it's not like a CCD that there's a crosstalk between adjacent channels. These are read out independently. It's a hybrid detector that has no sort of blurring, no point spread function. So these are phenomenal detectors. And what we've been exploring with the people on the I-24 beamline is how we can then put this together with developments in crystallization technology to see if we can solve structures straight from diffraction plates in a routine way. So there we saw a robot putting a diffraction plate, a crystallization plate on to the beamline. Here we can see a still of this. The X-ray beam comes through here. We can translate the plate to locate individual crystals. And by working with the plate manufacturers, we've now got crystallization plates that are very much lower X-ray scatterers than the usual plates. So this means that the background scatter from the plate can be really pushed down. And then if you, this as well as the X-rays coming through here, this is also the lens that we view what we're looking at through. It's a microscope objective. So if you look through the microscope now, the red square is a 20 micron beam of X-rays. These are crystals of a bovine enterovirus. They're about 50 microns across, and the 20 micron beam is going through it's illuminating these crystals, and they're being destroyed by the high intensity beam. So there's about 10 to the 12 photons a second into a 20 micron beam producing this effect. The lifetime is about 0.4 of a second. Because of these fantastic detectors, we can then collect 4 frames before the crystal blows up. And we just move on to the next place in the crystal, or the next crystal. And from a single plate, it was possible to solve the structure of this new virus. And the diffraction data are of extremely high quality. The electron density map is really beautiful, and the data are very reliable, as judged by the very good R factors in refinement. These R factors are not the same as the previous R factor. These show the match of the model to the data and are always higher. So these technologies already work and allow us to solve a new virus structure from a single crystallization plate using data collected at room temperature. That's the structure. We need technologies to improve because although the number of X-ray structures being deposited goes up sort of with a doubling time of about every 5 years, if we take viruses as an example of a much more difficult class of structural problem, a macromolecular complex, the situation is a bit worrying. In the early days, there was an increase. Now, it's not clear that in the last few years that we've really been able to sustain that. So there's a real value, I think, in developing the techniques to open up these important, difficult problems. And now I'll switch to talking about viruses for a few minutes. What we're trying to do is, firstly, understand the virus world and how it's evolved, how it's organized, and we've also got an ambition to try and do something useful if possible. And vaccines are a way of really, can be a very effective therapeutic route to tackle viruses. Smallpox has been eliminated for some years. Rinderpest, it was announced a few months ago that rinderpest, a very important disease of livestock, has been wiped out. So vaccines can work, they can eliminate viruses. The first structure we did was foot and mouth disease virus. Foot and mouth disease virus is still endemic, that's the red bits, in lots of the world, especially these tropical regions. And the vaccines that are available have had no lasting success in moving towards eliminating the disease. It's a very important disease economically. The vaccines that are administered, and here, this is a little movie I took on my iPhone earlier in the summer in South Africa of cattle being immunized. There's major and really relatively successful programs, but the problem is that this vaccine, well, apart from the problem the cow wants to get away, there's the problem the vaccine only lasts about 6 months, and then the protection is gone and you have to revaccinate. So the vaccines are not as good as they should be. So we determined the structure years ago of this, and then some years later there was a huge outbreak of FMDV in the UK, and it cost the UK billions of pounds, and the only thing that people could use the structure for was pictures on the front of the newspaper. It had no impact at this point on tackling the disease, so it's very disappointing. But if we look at the virus, we know that the problem with the vaccine is that the capsids are not stable. So what we want to do is to try and increase the capsid stability to give a more effective vaccine. And that will really solve a lot of problems about cold chains and giving the vaccine. It should also give increased lifetime if we have a higher titer of antigenic capsids. If the capsid breaks, it's no longer able to raise protective, it can no longer provide protective immunization. So if we look at the assembly, these are robust, this is the weak spot. Cross here, the virus falls apart very readily. So we must be able to surely use the structure to address this. One early idea was to replace these side chains here with cysteine residues that can form a covalent crosslink. And using the technology I24 I just described, very recently this mutation has been put into empty capsids of the virus, and the blue structure shows the disulfide cross bridge across the fivefolds. So this has been achieved, and the virus is indeed more stable, or the shell is indeed more stable. This is okay if we don't need to make a live virus. There's different ways of making a vaccine. All of the current vaccines rely on live virus. In the long run, what we want to do is to move to this sort of solution, get rid of the RNA, use a recombinant system to make empty capsids and vaccinate with those. And that should be a really safe vaccine. But that would take many years to get through the licensing procedure. So we're now working with a number of groups and a vaccine company to produce modifications to existing vaccines to improve their effectiveness. And I'll skip that because of time. Oh, so what we've been doing is, to use computer simulations, and I won't go through the details, it's using a standard molecular dynamics program, but with adaptations to allow us to get reliable results on what's going on at the interface between the two pentamers, the building blocks of the virus. And then we can use a standard thermodynamic trick to allow us to, from this single simulation, this one here of the full interface, to take snapshots of the dynamics run, unpick from each of those snapshots what the strength of interaction, what the energy, what the energetic terms here, here, and here are, and then calculate using general solvation models these, and hence calculate the strength of affinity, the strength of binding between these little triangular building blocks. That's then a measure of how strong the capsid is, and we can do this from this mechanism. Question is, does it work? We've produced calculations which show that a number of mutants, these are sort of being trialed by the vaccine company, so I haven't said what they are, but the sort of thing you'd expect. We can see that, for instance, the mutant B looks more stable. Several of these are significantly more stable than the wild type. So these mutations, the ones that look promising, have been put into viruses, and the one that I showed, mutant B, produces empty capsids that are stable for 2 hours at 56 degrees, when the normal wild-type virus is completely destroyed. And this is a fragmentary justification of that. It's a sucrose density gradient just to show that they're still intact particles. You'll have to believe me. And in fact, the remarkable thing is that most predicted stabilization changes work here. Not just one, but most of them do what they should do. They produce a more stable virus particle, and also they don't knock down the production of particles. They seem compatible with making large amounts of particles. So, and none of those were covalent modifications. They should, and the viruses are still capable of reproducing quite effectively. So I think very simple methods like this should in the future allow us to try and solve some problems with virus diseases where there are existing but imperfect vaccines available. And we're just starting work on polio as something where there's a real need to nail the problem. So I'll just finish by saying, can structure give us a grand vision of the virus world? Viruses, there's more viruses than there are everything else put together, multiplied by 10. They're hugely dominant, 10 to the 31 in the biosphere. There's a lot of different sorts of viruses. They infect everything. Their sizes, shapes vary, their molecular biology varies. You can't classify them easily because the rate of evolution means that the genome and the protein sequence changes very quickly. So within a family, we can understand the evolutionary relationships. Between families, it's impossible using the standard methods. However, if we look at the structures of the capsids, what we find, somewhat surprisingly, is that we can say something useful. We know about 80 different icosahedral virus structures at the moment, a tiny fraction of the total, but we've still got some useful information. And the sort of message from this is that, in fact, horizontal gene transfer, which you might expect to make it impossible to ever make an evolutionary tree for viruses, doesn't seem to operate at the level of virion architecture and assembly. So it seems that the virus has some sort of self-property, which is the shape of its capsid, that is preserved and transmitted vertically, allowing you to infer an evolutionary path between viruses over very massive lengths of time and variation. It's not true of most of the nature of the genome isn't so reliable, and the host interactions are completely misleading if you try and use it to classify viruses. So that's fine, but we can determine the structure of icosahedral viruses using electron microscopy or X-ray microscopy. But we have much less information about pleomorphic viruses, these baggy things here that have random shapes, and indeed, we don't know that much about helical viruses. So, therefore, it's really good news that despite the limitations, and in the face of only having a small number of structures, almost half of the recognized virus families can be classified into sub-4 separate lineages. So we can start to now get a grip on this. So we've got 4 lineages of viruses that are distinct from each other. This is a structure-based evolutionary tree. And these 4 lineages are so different from each other that we propose that they are polyphyletic in origin. So we don't think that they come from the same origin. We think they evolved independently. But within each of these branches, each of these lineages, you get examples of viruses that come from archaea, bacterial domain of life, and the eukaryotic domain of life. So the natural interpretation of that is that the different lineages of viruses are polyphyletic, but their origins are very ancient. Before the domains of life separated, there was already a separation of these viruses into their different lineages. There's a very poor fossil record for viruses, so we can't be certain of this, but this is the most natural interpretation of the data. And if we now look at this lineage-based classification, and we color them according to the four different lineages according to different colors, we can see that we can map out quite a lot of the traditional view of the virus world from the International Committee on the Taxonomy of Viruses, but you would need to restructure the traditional taxonomy to make it coherent with the structure information. So I think I expect over the next few years that we will have a slight shift in the way we see the virus world and the way we see the relationship between different families of viruses based on structure. Just to finish, the question, an outstanding question is, for a virus that's very large and not icosahedral, like vaccinia virus, can we say anything about the evolution of such a virus? If we take this lineage here, which includes adenovirus, PRD1 and PM2, famous because of Nicola's involvement in them, many of these are well characterized structurally. We know the key feature of this lineage is a double jelly roll in the major capsid protein. And this is PRD1. This is a trimer of the major capsid protein. And the PRD1 provided a nice explanation for how this type of lineage can make very large capsids using a tape measure protein to control the assembly of a very complicated system. What we've done recently is to look at vaccinia virus, which has about 200 genes. We've taken this point in this is the viral morphogenesis pathway. If you look at this point, the particle appears almost spherical. And this is deep etched electron microscopy. You can see it's almost spherical. You wouldn't say it's icosahedral, but it might have an icosahedral character, certainly much more regular than the final product. This has a scaffolding protein on the outside of a membrane that defines this spherical particle. This scaffolding protein is shown here, and Mohammed in the lab with others have done the structure of this protein, and In fact, it is a double jelly roll structure. So this virus fits, and if we look at the last slide, shows the way that this vaccinia protein fits into the evolutionary tree for this lineage of viruses that includes PRD1 and adenovirus. It fits in very comfortably. It's not an outlier. It's closer to the rest than, say, adenovirus. So this is very interesting because this protein is only present in the early stage of morphogenesis of the virus. And I think it gives us some hope that we might be able to classify even the most complicated viruses and the most fluid of the viruses if we can find a key point in their assembly pathway where they recapitulate their evolution. So for the complex viruses, we can argue, at least on the basis of one example, that virus morphogenesis, in the same way that for complex organisms, embryogenesis does the same, it recapitulates the evolution and allows you to sort of get a glimpse back into how an ancestral virus could have assembled, and a glimpse back to an ancestral time when the precursor virus had icosahedral symmetry. And I'll thank you very much.
You mentioned that advances in machinery and things, these are instrumental to advancing the structure in the correlative microscopy. You mentioned chemistry. Yes. In your mind, what would be the ideal tag to be putting onto proteins to make this happen?
I don't know. I'm not a chemist. That's why I blame the chemist for having to come up with the idea. I think ideally what you'd like to do is to have the fluorescent tag combined with something that was sufficiently electron dense that it showed up in electron microscopy. And that, I think, is probably a real challenge because you're likely to quench the fluorescence, and I'm sure there's all sorts of reasons why it's impossible. But I think there is the opportunity for progress if people work in that area, and it would be. At the moment, we have nothing that allows us to do that really. So it's a whole area that needs looking at, I think. I mean, the fluorescent tags are fabulous, but they only take you so far. They allow you to see where the tag is. If you can then get a link into something like electron microscopy, where you can see the actual structures and see the nature of what that tag protein is interacting with, then it does open up a whole new area, I think, of understanding.
I missed something in your comprehensive view of visualization, something about the disorder, macromolecular disorder, and where would you put techniques like small X-ray, small angle X-ray scattering into this panorama?
Disorder.
Macromolecular protein disorder, the implicit disorder.
Disorder, I said that the problem isn't three-dimensional or four-dimensional. You could say that a fifth dimension is disorder order, because the systems that we're talking about are not rigid. And that, as you know very well, many of the proteins are going to be disordered, at least in part, until they form an interaction. So that's a whole new dimension that I didn't talk about. But I suspect that the protein that determines the size of the PRD1 virus is exactly like that. It'll be just disordered, and then it will act as assume a structure when it assembles with the virus. Yes, I mean, you could stand up and give another talk on that, I'm sure. So it's a very interesting problem. But essentially, and that's part of it's why it's not enough to just do individual protein structures. We'll never understand the thing, because the whole molecular recognition and restructuring of shape when things recognize each other is fundamental to what we need to understand.
I have a question, Dave. In your talk, there is no mention about envelope viruses. What's going on with the envelope viruses?
Well, I mean, you could say vaccinia is an envelope virus.
Yeah, yes, but in terms of structure of an envelope virus, the full virus.
Yeah, well, yeah, I look forward to seeing the structure when you produce it, Nicola. I mean, as you know, electron microscopy has got some way towards getting, almost atomic structures. But crystallizing viruses with a membrane is very difficult, and we're at a very primitive stage in what we're doing, in my view. If you look at what's what's accelerated the production of protein structures, part of that is the protein production mechanism. You can clone these things, you can express them, you have a series of generic tags that are not dependent on the protein that allow you to purify it to high level. We don't have that for viruses. At the moment, virtually every virus has its own purification method that has to be worked up. We don't have things like tags, we don't have generic methods. Most of the methods for purifying viruses involve putting them in ultracentrifuge and sort of flattening them against something. So I think the thing there was a fantastic structure for adenovirus, as some people will have seen, that came out a few months ago from Hangzhou in California, which was essentially almost atomic resolution EM of this massive virus. But the thing is that adeno is very rigid. So you can make very good samples. The thing that's limiting with all of these techniques that's holding us back is sample preparation for the most part. And I think if you can make generic ways of purifying very fragile virus particles, that must also have value for other macromolecular complexes. I think there's a lot of work to be done in that area as well.
If there are no other questions, I will thanks again David for his talk. Thank you for coming.